Gut microbiota research is a booming scientific area, and its relevance for gastroenterologists and their patients can hardly be overestimated. The lay media are paying increasing attention to the topic, so gastroenterologists should be prepared to answer their patients' questions in a well-informed manner. This e-learning course aims at providing gastroenterologists with sound scientific information on the biological characteristics and mechanisms of the gut microbiota, as well as on its various clinically relevant aspects. At the end of this course, you will be able to advise your patients on all gut microbiota-related concerns. And you will know when the gut microbiota can provide leverage to improve intestinal health. To many people, among them even doctors, the term gut microbiota still sounds uncommon. But there is good reason for a change of names, as the old term gut flora implies plants, which is fairly misleading if we look at the reality of the microbial world within our intestines. As a consequence, the term gut microbiota has been established within the scientific community and is about to replace the old gut flora. Still, some terminological distinctions have to be observed. In Europe, the term microbiota is mainly used to refer to the global microbial population of the intestine, including the genes. A second term, microbiome, is used to refer specifically to the genome of this population. However, in other parts of the world, mainly North America, microbiome is simply a synonym for microbiota. On the basis of scientific publications cited in PubMed, the terminological change becomes visible. Over the past decade, gut flora has nearly been replaced by gut microbiota, or gut microbiome. The slide shows a number of microorganisms you probably know from your medical practice. What they have in common is that all of them are pathogens. However, they represent only a tiny part of the huge variety of species that form the gut microbiota, with many beneficial microbes among them. Now let's have a look at 3 common diseases the gut microbiota is involved in, albeit in different ways. Gastric ulcer, depicted by the left-hand section, is induced by toxic strains of Helicobacter pylori. Hence, in this case, a specific pathogen has been identified as a cause of a classical disease. For this achievement, Barry J. Marshall and J. Robin Warren received the Nobel Prize in 2005. By contrast, small intestine bacterial overgrowth, SIBO for short, provides an example for a condition that is caused by a local microbial imbalance, in this case bacterial overgrowth in the ileum. SIBO can be diagnosed through a breath test. The third disease, hepatic encephalopathy, develops through interference of the gut microbiota with the gut's global metabolism. Hepatic encephalopathy can be induced by the production of too much ammonia through protein catabolism. Another potential cause is the incapability of the liver to metabolize the ammonia into urea. The composition of the gut microbiota changes along the digestive tract depending on local conditions such as pH, amount of oxygen, kinetics, character of the mucus and secretions. The density grows on the way down, with the most abundant concentration to be found in the colon. The revolution in gut microbiota research has been triggered by new analytical tools, allowing for an in-depth investigation of the multifarious community within the human intestine. Advanced methods of gene analysis have overcome the burden and limits of culturing microbial samples. The facts that the international microbiota research community has gathered so far are really impressive. The gut microbiota, harboring more than 1,000 bacterial species, weighs between 1.5 and 2 kilograms. It consists of 10 times more cells than there are cells in the human body. The gut microbiota's metagenome, the so-called microbiome, is 100 times larger than the human genome with its 23,000 genes. thus presenting a much greater diversity. It does in fact form a second genome within the human gut. These microbial genes undergo frequent and rapid changes, which correspond to alterations of the gut microbiota composition. These processes are in contrast to the relative stability of our human genome. There are two ways to look at the gut microbiota. Firstly, which are the organisms that form this intestinal community? Secondly, what does it actually do? Let's turn to the second question first. The gut microbiota is involved in numerous functions of the body, particularly those that are part of the gut physiology. Gut microbes help feeding enterocytes and modulating the gut barrier. They interact with the immune system, they're involved in digesting and fermenting food, and in stimulating the local nervous system. In a number of trials, germ-free animals, that is, without gut microbiota, have been compared to conventional ones. The results show the importance of the gut microbiota for gut physiology, as well as for the general bodily comfort. Here are a few examples of the gut microbiota's various activities. It increases the enterocytes' turnover and feeds the gut epithelium mainly with butyrate, a short-chain fatty acid produced by fermentation. It increases the thickness of the lamina propria, and it also contributes to keep the local mucosal inflammation at a physiological level. It stimulates the nervous system, its sensory part as well as its motor part. It helps the immune system to mature and contributes to its maintenance. And, last but not least, it forms part of the barrier that protects the human host from the invasion of ingested harmful microbes. The gut microbiota has co-evolved with the human host and thus has adapted to our diet. Its microbes are called commensal, which means they feed on the same diet. The gut microbiota is involved in the processing of nutrition and it modulates the energetic value of the diet. Its enzymatic activities also influence the so-called bioavailability. This is the extent to which a nutrient or a drug can be used by the body. However, the host and the gut microbiota, although being commensal, do not feed on the same components of the diet. Instead, they perform a very useful division of labour. The gut microbes feed on what is normally referred to as non-digestible by the host. They also recycle substances such as nitrogen, sugars or fats that have escaped digestion, as well as residues from desquamated cells. Also, some B and K2 vitamins are produced by gut microbes. Interestingly, the gut microbiota is able to deconjugate many compounds which are conjugated by the liver, thus modulating the enterohepatic cycle and the detoxicative functions of the liver. In terms of co-evolutionary development, we can picture the human gut as an originally sterile host that has been colonized by a vast number of immigrants. This process has ended up in a largely peaceful coexistence. The host has learned to live with a selected gut microbiota at the price of a moderate stimulation of the local and global immune systems and with local inflammation that holds the normal microbiota at bay. It prevents it from invading the host without overstimulation of the immune response, which would be very costly for the host. This coexistence can be defined as an oscillating equilibrium that should be maintained as any major perturbation generates local, regional, and even global pathologies. The indigestible parts of the diet, the part not digested in the upper digestive tract, mucus and residues of desquamated cells, generate residues that in turn are substrates for other microbes in the colon, providing an amount of energy with an estimated 5 to 15% of the daily energy influx. Part of this energy is produced in the form of short-chain fatty acids that provide fuel for epithelial cells and for the lipogenesis among the linen helix. Another product of fermentation are gases. Their volume amounts to an estimated 10 litres per day, consisting of CO2, H2, CH4, H2S, and other volatile compounds. The gut microbiota's energy output is even more impressive in mice. A germ-free mouse needs 30% more food intake than its conventional counterpart with a gut microbiota. From all this, the importance of the gut microbiota for energy balance, as well as intestinal health and comfort, becomes patently clear. One of the most exciting research areas concerns the relations between gut microbiota and the brain. Meanwhile, there is a considerable body of evidence suggesting that the gut microbiota modulates certain brain functions, which in turn influence behavior. The gut microbiota, and the brain are connected through numerous pathways, among which the vagus nerve is one of the most important ones. The diagram shows all these functions in separated ovals in order to provide a clear overview. But in reality they overlap, together creating a homeostatic balance. This balance is permanently oscillating between the poles of health and disease, with some in-between states that might or might not be associated with symptoms. In the third part of our e-learning course, we will be concerned with this topic in a more detailed manner. However, there is still the chicken or egg question. Does the disease change the microbiota or vice versa? This entails another question. Is it possible to restore a healthy gut microbiota in order to manage certain diseases? We will get back to this issue later on when dealing with the role of probiotics for intestinal health. To explore the gut microbiota, different tools can be used. One is bacterial culturing, using various media to distinguish and identify different sorts of microbes. The other one is genomics. Both methods can be considered direct tools for detecting which kinds of microbes live in the gut and what functions they have. The stool culture test is used for the detection and identification of pathogenic bacteria in the stool. In the laboratory, small amounts of fresh fecal samples are being streaked onto different media. The diagnosis is based on phenotypic characteristics such as shape, gram sensitivity, and biochemical specificities. Culturing, although a well-proven technique, has its limitations, as by far not all microbial strains can be cultured. Genomics is suited to overcome this bottleneck. By applying this tool, we have learned, in particular by identifying 16S ribosomal RNA, that the human gut harbours a lot of anaerobic microbes that have not been detected by culturing. While one human stool sample contains about 100 to 150 microbial species, the number of species actually living in the human gut is an estimated 500 to more than 1,000. The major phyla are Bacteroidetes, Firmicutes, Prevotella actinobacteria. The specific profile of the individual's gut microbiota is to be found at the strain level. Every human has specific bacterial strains, similar to the individual patterns of fingerprints. Gut microbes are classified into the following taxonomic units, which follow a top-down direction: phylum, order, genus, species, and strains. To take an example from the animal domain, one phylum would be the chordates, which cover a broad range from bear to snake. A corresponding order would be the carnivores. A genus belonging to the carnivores would be bear, while the polar bear would be a species of the bear in general. One step further down the taxonomic ladder is the strain. In our example, a strain would be the polar bear in the nearby zoo. Therefore, the difference between, say, Bacteroidetes and Firmicutes are similar to those between a bear and a sea star. This and the following slide show 5 bacterial phyla plus the Archaea phylum, whose members are important for gas production. Regarding E. coli, you might be familiar with the following strain: ETEC 0157H7. Let us take the species E. coli as an example. What you see at the bottom of the diagram is this specific strain, which is named by a code, ETEC 0157H7. By applying genomics, that is the sequencing and analyzing of genes, we can gain a whole lot of different kinds of information. Analyzing 16S ribosomal RNA unveils the identity of the microbe. Other parts of the RNA tell us which proteins are being synthesized. In turn, looking into proteins provides information on the enzymes or messengers that have been produced. And the investigation of the DNA brings to light the genetic constitution of individual microbes as well as of whole microbial populations. Having touched upon direct diagnostic tools, we now turn to indirect tools of gut microbiota exploration. 2 kinds are to be distinguished. Firstly, the detection of gas that some microbes produce when fermenting adequate substrates. Secondly, the detection of specific antibodies circulating in the blood. One example is the urea breath test, which is simple and non-invasive. Urea breath tests have been employed to diagnose H. pylori infection. The test makes use of the fact that H. pylori produces large quantities of the enzyme urease. 13C and 14C urea breath tests, the C stands for carbon isotopes, offer an excellent diagnostic yield. Patients ingest a solution containing 13C or 14C labelled urea. Then, when exhaling, their breath is sampled for isotope-labelled CO2, which is released by intragastric H. pylori urease activity. The test can be completed within 20 minutes and is highly sensitive and specific. The same principle is applied to diagnose small intestine bacterial overgrowth. Here it is an early increase in hydrogen concentration in the breath after lactulose ingestion that is indicative of the disease. We have reached the end of the introductory part. By now you have a rough overview over the field and its huge relevance for gastroenterological diagnosis and therapy. Would you like to learn more about the gut microbiota's medical potential and in greater detail? Then you might want to proceed to Part 2. Its focus is on the question when doctors should care about the gut microbiota of their patients.